So in this video we are going to discuss about the muscle contraction theory or sliding filament theory. First of all, we are going to discuss about the skeletal muscles, which works under voluntary control and are composed of bundle of muscle fibers. Muscle fibers are the long cylindrical cells containing several nuclei. Muscle will contract or relax when they receive the signals from the nervous system. A neuromuscular junction is the site of signal exchange. This is where the synaptic bulb of an axon terminal and the muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomere, which run adjacent to one another down the length of the myofibril. Each sarcomere consisting of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere called amyline, and the thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filament sliding along the myosin filament. Although the action between the filament is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridge of the myosin filaments attach to the actin filament and exert a force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomere shorten without the thick or thin filament changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate, causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered along the myosin to pull the actin filament toward the amyline, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. Myosin remain attached to the actin until a new molecule of ATP binds. The myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscles to relax. Muscle contracts are controlled by the action of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross bridge binding site on actin, and when calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces the tropomyosin. Exposing the myosin binds site on actin, this allows myosin to attach to a binding site on actin filaments, a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in the response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to a receptor, which depolarizes the membrane of muscle fiber. The electrical impulses travel down the T-tubule and the muscles slide along each other. The entire sarcomere shorten as the Z lines draw closer to the amyline. As the sarcomere in myofibril contracts, the entire muscle fiber will shorten. When muscle fiber contract in, a muscle can produce enough force to move the body and allow you to do the daily activities in your life.